All right. This is about where we left off, and it's a question involving the layers of the epidermis. Now, remember, the epidermis is the outermost layer of your skin, and it gets subdivided based on the arrangement of cells. The listing that I have here is starting, you can see 1, 2, 3, 4, 5 layers, with the deepest layer, because I kind of look at this as a journey of the keratinocyte. Keratinocytes are the dominant skin cell. They're a kind of squamous cell, and they're born in this basal layer. Its full name is stratum basal, as you can see. A lot of mitosis here, and it used to go by an old name. You might see it around in some old textbooks called the stratum germinativum, okay? Because germinal, when you use the word germinal, it means reproductive. So anyway, it's an area with a lot of mitosis. You make a lot of new keratinocytes, and you're doing this all the time. And what happens to them is they grow, and as they grow away from this layer, they become part of the stratum spinosum. So this is a thick layer of living cells. The keratinocytes at this time are making a lot of keratin, which is the protein that kind of defines them. That's why they're called keratinocytes. It's a strong layer for your skin, and it's also a place where melanocytes, which are scattered in that basal layer, will start to distribute the pigment melanin to the keratinocytes. So, for example, one of the things to remember here is that when you add up where most of your melanin is at any given minute, it's in your keratinocytes mostly. Even though the melanocytes are the cells that make it, it's the keratinocytes that are acquiring it and spreading it out through the skin, so you can have a kind of even layer of ultraviolet light protection with that pigment. The other cell that you'll find here and there in your stratum spinosum is called a Langerhans cell. This is, of course, named after a person who discovered it. What it is a white blood cell, rather like one of the big macrophages that would float around in your bloodstream. This is a white blood cell born in your bone marrow, travels through the circulatory system, and then kind of wriggles its way out of the blood vessels in the dermis and works its way up into your skin, your epidermis. And it's there to help protect you. It only lives a couple of weeks, like most cells in the skin, and then it is replaced by another over time. Now, the cells in the spinosum keep being pushed upwards by the reproductive rate in the basal, and so eventually they become part of the stratum granulosum. And this name is again based on appearance. So the cells at this layer that are keratinocytes flatten out, and they do indeed look like what they are, squamous cells. See, it's not a real thick layer, right? But when you stain it, you see little granules. At least that's what people called them. What's happening is the keratin inside the cells and the melanin is picking up the stain, and it looks like there's little graininess to the cells. Now, after the granulosum, the cells are too far away from the blood supply, which is back in the dermis. Okay, so at this stage, their life is pretty much over, and they will become part of the dead layers, which are okay in the sense that they're helping protect your skin. A layer or two of dead cells on the outside of the living is just a method of protection. Okay, now when a keratinocyte dies, it leaves behind the protein keratin. And so that's kind of what you would see if you ever look at up close a set of dead skin cells. All you get is like the protein remnants. Okay, so it's not very exciting. The stratum lucidum is the first dead layer in thick skin. Now, thick skin is palms and soles. Any other skin is thin. Okay, so this is only a layer we find with the thick skin. However, whether you're thick or thin, you will have a stratum corneum. Okay, corneum means dried out. So this is just old flaky, you're kind of losing this stuff everywhere you go. Humans shed their skin constantly because there's always this turnover. And so our old dead skin becomes the corneum, and the corneum peels off a little bit at a time. But while it's there, it's helping protect the living cells underneath. Okay, now some questions to go along with this discussion of the epidermis. Question 28, how do keratinocytes respond to mechanical stress? I mean, it sounds like a weird question, right? But here's where I'm going with this. If you do something with your hands, for example, like a lot of people work with their hands, the skin on the palms of your hands will get thicker. And that's a response of the keratinocytes to being mechanically stressed. So, you're working in the garden a lot, or you're doing a construction thing, or you're at the gym lifting weights. All that working with tools is tough on the skin, and it's putting forces through the skin, which creates higher rates of reproduction in the keratinocytes. And so they get thicker. And so if you've ever had a callus, this is exactly the kind of thing that I'm talking about. All right, now another thing that happens, it's not like keratinocytes have a lot of, repertoire in their behaviors, but another thing that can happen with keratinocytes is you could separate them from their surrounding keratinocytes, and they don't like that. What we're really talking about here is a cut or a laceration or some kind of skin injury. And so when that happens, they do the exact same thing. They increase their rate of mitosis. So notice whether you're stressing them mechanically or you've separated them from each other, they respond by, reproducing faster. In the one hand, with the mechanical stress, you're trying to build up a thicker skin. When you're talking about a cut, what you're trying to do there is fill in the space. So that's what we call a healing by primary union. You have a scar tissue at the base of the wound trying to pull the edges together while the keratinocytes are reproducing like crazy to try and fill in the space. And then finally, describe what happens to keratinocytes as they grow up toward the surface. So this is kind of the journey we just described as you go from stratum basal to stratum corneum. And it's about a 2-week journey for skin cells. And during that time, what they do is they begin by cell division. They make a lot of keratin. They pick up melanin from the melanocytes and make sure it's distributed around. And then they flatten out. Remember, that's in the stratum granulosum layer. They actually look like squamous cells there. And then finally they die. And that's their little story. Some cells that are associated with the epidermis. What's a Merkel cell? You can see an illustration over here. It's this little strange looking thing at the base here. This is in the stratum basal. That is the most superficial of all skin sensory receptors. We think it's tied into light touch or fine touch sensations. Okay. And then you've kind of heard this one already. What's a Langerhans cell? It's this kind of octopus looking cell here. This is a white blood cell, basically a monocyte which was converted from a macrophage. So it's a big cell of the immune system that has the ability to phagocytize and engulf, invaders. It's here to protect you. For example, if some bacteria or something's trying to get through the skin, your Langerhans cell will sound the alarm and start the defense. Okay, now this sounds like connective tissue questions, right? Except that, well, we got to talk a little bit about the dermis here. This is the layer below the epidermis, which is what we've been describing. And it has 2 sublayers, okay? The more superficial layer of those 2 is the papillary. Okay, now you remember me seeing this picture before. This is loose connective tissue. There's a bunch of little fibrocytes and fibroblasts scattered around the cells. And then the protein fibers that you can see that have been stained are the thicker collagen bands and then the little spiderwebby looking elastin fibers. Loose connective tissue because it's not tightly packed. And this is what you find in the papillary layer of dermis. As you go a little bit deeper, below the papillary layer of dermis, you have this really built strongly connective tissue. You can see bundles of collagen kind of swirling in all different directions. You can also see a handful of little darker stained fibrocytes and fibroblasts. This is the kind of connective tissue you find in the reticular, which is the deeper layer of dermis. So this is dense, irregular connective tissue. The glands that are associated with your skin, of course, exocrine glands, there will be different ways in which they secrete their substances. And so there are three methods of secretion. The one I have illustrated here is the merocrine. Okay, and a good example is just plain old sweat, what we call eccrine sweat. It's kind of salty water. It's used to cool you off. And the cells don't undergo any drastic change. They just secrete little droplets of sweat. That's a merocrine method. Now the apocrine method, which can be used by some kinds of sweat glands, we literally call them apocrine. They're the sweat glands in your armpit or your anogenital area. But really more widely known as a type of secretion used by the mammary glands. And so when these kinds of cells secrete their product, they lose little pieces of their membrane. It's a little bit destructive, this process. And so I've tried to illustrate that with, instead of just plain old colored bubbles coming off the cell, you see how they're wrapped in a darker membrane like the cell membrane. So what's happening is the cell is undergoing a little bit of damage as it releases its product, in this case milk. And then the most drastic method of all is the holocrine method, which is utilized by the sebaceous glands of the skin. These are the oil glands. And what happens here is the cell disintegrates. It literally bursts and releases all the oil. And that's part of why oil is well known to clog pores. It's always got debris in it. Bit of a list here about the functions of the integument, and obviously I talk about this in the lecture too. What you'll notice is the first three things noted are barrier, different kinds of barrier, right? So the chemical barrier is because you secrete oils onto your skin, which are fatty acids, and the pH is a little bit lower than neutral. It's like 5.5, and that does help reduce the growth of microbes on the skin. It doesn't do a lot, but it does something, okay? The biological barrier is the Langerhans cells. That's your immune system being kind of implanted inside the skin. The physical barrier is that the skin is thick. It has multi-layers to it. There are things that can go through it because, as you know, cells are bordered by a cell membrane, which is made of lipids. So things that are lipid soluble, and I have a few listed over here, things that are lipid soluble will go right through it. Oleoresins, that's actually a reference to the urushiol, which is a resin created by poison ivy, which poison ivy is not really that poisonous. It's just some people are allergic to it. So it soaks into your skin and you have an allergic response against your skin that's been contaminated by the urushiol. Solvents, organic solvents, my advice is always, don't wash your hands with paint thinner. Paint thinner is for paint. It's not for people. This will go right through you, and it will damage your liver and your kidneys and, you name it. And then salts of heavy metals. This is not as big a concern as it might have been during more industrialized times here in America. But people do occasionally get exposed to heavy metals. They really, nowadays, most of the metal exposure people get is from contaminated drinking water or contaminated seafood or something like that. Now, this is quite an important one, number 4 here, temperature regulation. And your skin is a hugely important, actually, organ in this process. The sweat glands, when sweat evaporates, it carries heat with it. So this is the way we mostly cool ourselves. You may have heard of conditions like heat stroke, and what happens there is you kind of lose the ability to sweat. You sort of run out of the way to do this, and that's when people overheat and go into what's called a malignant hyperthermia. What I would tell you about it though is that heat stroke is 100% preventable. You just give people water or cool them off. You have to monitor them, but every year, People are out, doing things, and they get caught up in it, and athletes in particular. And you will hear about deaths from heat stroke in the summertime. Um, the other thing to think about besides the heat management is the cold management of your skin. So you have blood vessels in the dermis, and you can change their diameter. So let's say we'll go back to the heat for a second. The vasodilation means the blood vessels get bigger. So when it's really hot, you dilate vessels in the dermis of the skin to bring more heat to the surface so you can shed it faster, so you can get rid of it. Now, in the winter, in the cold, you're going to vasoconstrict, which is great for preserving the core temperature of the body, but not so great for your extremities because now they lose the protection of body heat. And this is why when people get frostbite, it's typically in toes or fingers or their nose or something like that, because those are the extremities that were deprived of adequate flow and heat. There is an additional danger, and I'll just tell you guys about it just so you'll keep yourselves out of trouble. If you are out in the cold and you're drinking alcohol, say at a football game, the alcohol produces a byproduct called acetaldehyde, and the acetaldehyde causes vasodilation. So think about that for a second. You're in the cold, and your body's trying to keep heat in your body, right? And now all of a sudden you're drinking something that causes vasodilation all over the place. So you're going to lose heat even faster. And this is how people get themselves into hypothermia, or it's one of the ways anyway. So be careful. Sensation, obviously an important part of the skin, and there are different kinds of receptors. For example, the Meissner's receptor is a light touch receptor. The Pacinian is more of a deep touch or pressure receptor. The root hair plexuses are part of free nerve endings, which are also involved in pain sensations. Heat is a free nerve ending consideration, whereas cold is its own kind of receptor. It's called a Krause's bulb, Krause, Krause's bulb. And then the other thing, when you add all this up, I'm going to be able to give you guys a kind of a descriptive pathway where we will connect these different kinds of receptors back into your brain. We're not really going to do that, though, until Unit 3, which is part of the nervous system. Metabolically, one of the things you can count on your skin for is to participate in the process of making your own vitamin D. It's a set of enzymes in your skin that are actually activated by photons, by UV light. So you got to get some sunshine. The blood reservoir consideration is kind of what I was talking about a little bit earlier when I was saying that the blood vessels in the dermis could vasoconstrict or vasodilate. So you can change flow of blood to the skin in order to preference blood flow to other organs. And then the excretory functions of the skin are not at all efficient. You can get rid of some metabolic waste by sweating, but you're not going to do it anywhere near as efficiently as the kidneys. And this is the comment I like to make here is that if, going to the sauna took care of all that, because that's kind of like a pitch people do for selling saunas. They'll say, oh, you go in the sauna, you sweat out the toxins. Yeah, not really. If that worked, then you would just put your kidney failure patients in the sauna. And we don't, because that's not it's not an acceptable treatment, and it's probably a terrible thing that would kill your patient. But yeah, I have actually seen kidney failure patients sweat out urea. It's a very interesting phenomenon, and it's exactly what you think. There's a urine odor to their sweat, and when it dries into a kind of a white powder. And I remember the first time reading about it when I was a student. It's called a uremic frost because they have this white powdery stuff on their skin, and it's really a dire finding because it says their kidneys have failed, and you either have to get them a transplant or you have to get them dialysis. But the sweat glands cannot compensate for what the kidneys do. Okay, a couple of short questions here. In a sample of skin, where is most of the melanin found? You might have heard me say this earlier. Even though it's made by melanocytes, the pigment producing cells, they share it with the keratinocytes. So most of your melanin is actually distributed through your keratinocytes. Question 38, what's the function of melanin? Blocking UV radiation. So it's protective against radiation damage to the DNA in your skin cells. So the more melanin you have, the less likely you get skin cancer. What is the connotation of cyanosis, erythema, and pallor? So these are color kind of descriptions that people say about skin. Cyanosis literally means you're turning blue, and it isn't because your blood turns blue. It's because your blood is deoxygenated, and when the pigment in your blood, hemoglobin, is not carrying oxygen, it's carrying carbon dioxide, it turns more of a magenta color. But with the skin over the top acting as kind of a filter, what we see is blue. And I will tell you, I have actually seen people turn blue. I treated a patient one time in the ER for status asthmaticus, which is a really bad bronchoconstriction event consequent to asthma, and she was blue. And so we had to, obviously give supplemental oxygen. You want to increase the intake of oxygen, and we had to break the spasm in her breathing passages, in her bronchi. And we were able to do that, but it was kind of a scary moment for sure. Now, erythema is a redness. Now, this is not blushing. I want to clarify that. Blushing is fine. It doesn't carry any bad connotations, but erythema is the word you use when there's a redness associated with inflammation. Inflammation is a response to injury. And if you've had a first aid course, maybe you've heard, the 5 cardinal signs of inflammation. It's redness, it's heat, it's swelling, it's pain, and it's compromised function or loss of function. So if you came in with a sore throat and I looked at your throat and it's all red back there, I don't write down in the chart red throat. I would write erythematous pharynx. And that's the jargon does make the difference. Okay, so this is part of what you guys are learning, how to say the things that are obvious, right? Yeah, it's a red throat, but don't write that. Write erythematous pharynx. Pallor is where you're too pale. Lots of people are pale, and it's fine. But when they get excessively so, we say that's a pallor. And it's an indication that there's insufficient amounts of hemoglobin. So their normal pinkish redness, whatever, is just not there. And it's a sign of what we call anemia. And anemia is a lack of red blood cells or a lack of the pigment hemoglobin. It is most often caused by bleeding, but there are other causes of anemia too, like nutritional deficiencies and things like that. Question 40, what are the differences between eccrine and apocrine sweat? You might have heard a little bit about me talking about this earlier. The eccrine sweat is everywhere, and it is salty water, and its major contribution to your health is to help cool you off. The apocrine sweat is also salty water, but you have added things like there's some fatty acids in there, there's some small proteins in there. There might even be some pheromones. There's a word down here for you. Could be a new word. Pheromones. Oh my, that's an email. The pheromones are interesting. Humans don't officially have any, but there's all kinds of compelling evidence that we do. My favorite bit of research involving the pheromones is they did a study in Los Angeles where they were looking at the phenomenon of how women, when they are housed together, will sync their menstrual cycles to each other. And the way they did it is they kept the women separated, actually. So they had one group across town, say, donating sweat samples from their axillary sweat, their armpit sweat, putting into little pads, soaking into little pads. And then they had another set of women across town sniffing those every day. So they would transport them, and then the other women would sniff them. And they had a control group of women that were just sniffing like vanilla extract, you know. But what happened is, over a relatively short period of time, the women who were sniffing the sweat from the donor women synced their menstrual cycles to the donors, which is tells me there's something in the sweat. Man, that would be a pheromone of some kind. Anyway, it hasn't been identified, but there's evidence of that. So it's interesting. And the biological rationale for doing that is supposed to be a kind of reproductive competition, so that women are syncing their cycles so they're not going to be outcompeted for reproductive success. The other things that you guys will see here that I've noted about apocrine sweat, making it different from eccrine, is that this is the one that's associated more with odor, because and in our society that means you need deodorants, right? Uh, there are things for bacteria to consume in apocrine sweat. Like, bacteria can't metabolize salty water. That's not really food, but fats and proteins, that's food for microbes. And so as the bacteria break these things down, they give off odors. Just some definitions that go along with studying nails, fingernails or toenails. And so an eponychium is the proper word for the cuticle. The perinechium is the skin along the sides of the nail plate. And the hyponychium is the skin underneath the free edge of the nail, which you may have clipped if you ever clipped your nails too short. This last thing to note is just a real simple line diagram picture of a slice through the skin, so you could identify different layers. You can see everything's been labeled here. And I just would encourage you to kind of do the picture a couple of times so you know the parts. The lower level, which is the hypodermis, then the dermis, and then the epidermis. There's some labeling here for sebaceous gland, which is an oil gland associated with the hair follicle. Little tiny smooth muscle, which can make your hair stand on end. It's called the erector pili muscle. And then what am I missing? Oh, the sweat gland down here, kind of on its own, and then the shaft of the hair as well. And I hope that helps, and that should wrap up our Paperchase for Unit 1. Stay well and study well.